Hello friends, my name is Dr. Sahoo and I want to study the hematopoietic system. Okay, the first topic we want to cover in the hematopoietic system is the TBW. What do you mean by TBW? TBW means total body water. Total body water is constitutes 60% of our body weight. 60%, that is being 60% of our body weight, and it includes ICF and ECF. ICF is intracellular fluid, that is the fluid that is being present within the cell and is constitute 40%, and another one is the ECF, it is the extracellular fluid that is constitute 20%. Again, the ECF is divided into interstitial fluid that is being 15%, plasma that is the 5%, and TCF, transcellular fluid. Interstitial fluid means the fluid that is being present between the cells, that is the interstitial fluid, and is constitute 15%. Again, plasma, what do you mean plasma? If we remove the elements from blood, that is constituted only plasma, that is the 5% of our total body weight. What do you mean by transcellular fluid? Transcellular fluids are synovial fluid or cerebrospinal fluid that is present within our brain. Okay, so we want to then study the measurement of the component. First we want to measure TBW, the total body water, and we can measure with the help of D2O and T2O. D2O is deuterium oxide and T2O is the tritium oxide. It is a heavy water form of heavy waters. Okay, and next we want to cover is the ECF and GFR. ECF is extracellular fluid and GFR means glomerular filtration rate that is being conducted in the kidney, and we want to measure with the help of inulin. We can measure it with the help of inulin. Again, plasma volume, we can conduct the plasma volume measurement with the help of Evans blue. Evans blue is a dye, and through this we can conduct the measurement of the plasma volume. Again, there will be a question, how can we calculate the intracellular fluid? We can calculate the intracellular fluid by TBW minus extracellular fluid. We can calculate TBW by D2O, T2O, and ECF by inulin. So if we subtract that component, we can got the ICF component, ICF measurement. Okay, so our next topic is the blood. The blood having a pH of 7.4. First, blood is a fluid connective tissue that is present within our system, and it is the pH is 7.4. First one, acidic component of blood is the venous blood. Venous blood is acidic because there is presence of CO2. As we take respiration, the CO2 being transported within our blood, and that way the venous blood contains CO2 maximum portion, so it gives acidic component in comparison to arterial blood. And next component acidic is blood cells is the acidic component. Okay, so the blood another component is the plasma that is being 55%, and another component constitutes the cells that is being 45%. The color of the blood comes from RBC, that gives red color to the blood, and the color of the plasma comes from bilirubin. The degraded product of the RBC is the bilirubin, and that gives yellow tints to the plasma. The cellular element of the blood are RBC, WBC, and platelets. Centrifugation, upon centrifugation of the blood, the upper layer will be the plasma, that is constitute 55%, and middle layer will be the WBC and platelet that constitute less than 1%, and less than 1% is the buffy coat layer, and lower layer will be the RBC, that is being 45%. The next first cellular component we want to study or teach is the WBC. WBC constitute of granulocyte and agranulocyte. The granulocyte is being called granulocyte because it contains granules, and it is also known as PMN, polymorphonuclear cells. What do you mean by polymorpho? Because it contains segmented nucleus or band form of nucleus. Whereas in case of agranulocyte, there is no granules are present, and it is called PBMC, peripheral blood mononuclear cells, because it contains only single nucleus without any segment. Then the granulocyte constitute neutrophil. The neutrophil is called neutrophil because it take off the neutral dye. In case of eosinophil, it takes the acidic dye, and in case of basophil, it takes acidic dye. And agranulocyte includes monocyte and lymphocyte. Then we will focus on the function portion. The neutrophil is highly phagocytosis and is the first line of defense in case of infection. Then next one is the eosinophil. Eosinophil number increases in case of allergic reaction, and in case of parasitic reaction. The parasitic includes nematode, flukes, cystode, and it inactivates the allergen. Here allergen indicates the histamine. Okay. Basophil, if basophil is activated, it secretes heparin. Heparin is an anticoagulant structure that maintains the blood fluidity. And the histamine is an allergen that causes vasodilation and many more inflammatory functions. Monocyte, if present within the blood, it is called monocyte, and if it escapes from the blood and comes to tissue, it is called macrophage, and it present in chronic inflammation. Chronic inflammation condition is TB. TB is a chronic infection condition, and it is the largest WBC. Monocyte is the largest WBC. Lymphocyte helps in antibody production. There are three types of lymphocyte. There is the B lymphocyte, T lymphocyte, and natural killer cells. Again, we will focus on serum. What do you mean with serum? If we subtract plasma, if we subtract fibrinogen from plasma, it is the serum portion. Band cells is immature neutrophil is the band cells. Segmented cells, mature form of neutrophil is the segmented cells. Macrophage, if the monocyte escape from the blood cell, it is called macrophage. Plasma cell, the activated B lymphocyte. The activated B lymphocyte is the plasma cells which secrete antibody portion. Okay. If we target on the size, the WBC has highest size, then RBC, and then platelet. If we focus on concentration, highest concentration is RBC within the blood, next platelet, and next WBC. In case of ruminant, pig, and poultry, the lymphocyte has highest constitute, that is a 60 to 65%. Whereas in case of horse, dog, and cat, neutrophil is the highest, that is a 60 to 65%. Okay. Then we focus on macrophage of different system. If the macrophage of blood, it is called monocyte. The macrophage of liver, it is called Kupffer cells. The macrophage of lungs is called Dost cell or alveolar macrophage. The macrophage of kidney is the kidney mesangial cells. The macrophage of brain is the microglia. The macrophage of the bone, it is osteoclast. And the macrophage of the placenta is a Hofbauer cells. So next topic is the lymphoid organ. Lymphoid organ is divided into primary lymphoid organ and secondary lymphoid organ. The primary lymphoid organ is that organ in which the lymphocyte get differentiate from the stem cells. It includes bone marrow, thymus, and bursa of Fabricius. The bursa of Fabricius is present in the birds. The secondary lymphoid organ is that organ in which the lymphocyte get exposed to antigens. It includes lymph node, spleen, and malt, galt also. The APC, what do you mean by APC? APC is the antigen presenting cells. The antigen being presented to helper T cells through APC. It includes dendritic cells, macrophages, and B lymphocyte, and it exposed to T helper T cells. Then the maturation site in case of B lymphocyte is the bone marrow. Whereas in case of T lymphocyte, the maturation site is being thymus. Immunity type. The type of immunity in case of B lymphocyte is the humoral immunity. Whereas in case of T lymphocyte, it is being cell mediated. The humoral immunity, it is being called because the B lymphocyte acts on the body fluid, in the blood, in plasma. And in case of T lymphocyte, it is called cell mediated immunity because it acts upon the intracellular pathogen. The secretory product, the B lymphocyte secretory product is the antibody. And in case of T lymphocyte is the lymphokine. The secretory antibody. What do you mean by antibody? Antibody, actually the B lymphocyte upon exposure to the antigen, it gets activated and divided into plasma cells. The plasma cells, the activated plasma cells secretes the antibody. Like the T lymphocyte secretes lymphokine. The lifespan of B lymphocyte is short. whereas the lifespan of T lymphocyte is quite long. Okay. Then after that we will study upon the different classification of immunoglobulin. How can we remember the immunoglobulin? Through a code word GAMED. IgG, IgA, IgM, IgE and IgD. Then the IgG, immunoglobulin G, it constitutes 80% and it crosses the placenta. It crosses the placenta. The statement is given in the recent 2023 ICR paper, ICR JRA paper. IgA is a 10 to 15% and it is a secretory type antibody. What do you mean by secretory type antibody? Secretory type antibody means it is being secreted from the epithelial lining and from mucus, from tear, from saliva. Okay. IgM, it constitutes the 5 to 10% and is a primary immune response. What do you mean a primary immune response? If some infection being occur, the primary immune response means IgM is first produced, and like that IgG is a secondary immune response. It is being secondary immune response. It is being secondary immune response to IgG. IgD constitutes the 0.2% at the surface present on the B cell surface. So it is also known as a B cell surface antigen, also called as. Again IgE is the 0.1% that is being least present, and it is been activated in case of allergic reaction, in case of parasitic disease, in case of hypersensitivity reaction. So this is the topic. So student, let's discuss some question related to the hematopoietic system. What is the normal pH of the blood? Option A 7.4, option B 7.2, option C 7.8, option D 7. The answer is option A 7.4. Okay. So the next question is, the blood volume of an animal is generally dex portion of its body weight. Option A 8%, option B 4%, option C 12%, option D 6%. The answer is option A 8% of its body weight. Then the next question, the Buffy coat on centrifugation of blood reflects. Option A WBC, option B RBC, option C platelet, option D both A and C. Answer it, comment down below. And another question, the naturally occurring antibody against RBC in case of blood incompatibility is. Option A IgG, option B IgA, option C IgM, option D IgD. Answer it below. Thank you.